Hi, this is Dick Osgood, Executive Director of the Lake Minnetonka Association. Welcome to the next installment of Lake Minnetonka Association Presents. With me today, at least with me in spirit today, is my friend and colleague, Dr. Dan Molloy, who is in hiding somewhere in New York. Welcome, Dan.
Thank you, Dick. And I'm in New York, but I'm not in hiding. Go right ahead.
Okay, well, we'll work on finding you, and you can work on finding yourself. But today's topic is the Zequanox trials in Lake Minnetonka. And Dan, you know a little bit about zebra mussels and Zequanox, don't you?
I'd say so. I've been working on them ever since they came into New York in 1990, doing research to find green control methods. In fact, the USGS project is going to evaluate this biological control method that I'm the patent inventor of. So it's pretty exciting.
Well, good. I'd like to start out. Dan is an expert in zebra mussel biology and ecology, and in Zequanox, the method that's being tested to control zebra mussels. But before I start that, Dan, could you give us a primer on zebra mussels and why they're a problem?
Yeah, if we could maybe take a look at slide 2. I sent you some slides, Dick, so I assume you can get them up there on the screen.
It's
and
there now.
See a hand, yeah, you see a hand there with some mussels in them. So anyone who has never seen a zebra mussel can get an idea that they're basically fingernail-sized critters, and of course the magnification in that photograph is maybe not great, but they do have stripes. That's why they got the name zebra mussels. And then if we go to the next slide, you'll see a zebra mussel, and you can see that there's some hairs coming out the bottom of it. Those hairs are the reason they're such a problem, because they're called byssal threads, and there's no other mussel in North America that produces threads like that. It allows them to attach to any hard surface. And there are probably about 30 or 40 hairs all matted there. It's a little wet, so they're not divided up so you can see them. There's no other freshwater mussel in North America that can do that, and that is one of their major biological characteristics that is so different from anything we've had before, and the nature of the problem of their fouling. If we go to the next slide, on the left, you can see me holding a native mussel that might be, 3-4 inches long, and at the top, you see a cluster of zebra mussels. This is one of the clearest ecological effects of zebra mussels coming to North America. They're killing off our native mussels, like you have there on the left. It's because literally that native mussel, that big mussel, does not have enough food and oxygen. It's being stolen by the mussels at the top. And on the right, you have a pipe that people have heard stories of getting clogged. So there are industrial impacts also. And if we go to the next slide, and this will be the final one in Zebra Mussels 101. On the left, you see a drawing, and hopefully it's magnified enough you can see something called the inhalant siphon with little tentacles all around the edge. And that's where the mussel takes in water. And then at the very top of that drawing, you see a label exhalant siphon. That's where the water comes out of the zebra mussel. And it's taking water in for food and oxygen. On the right, you have an actual photograph of a mussel that's opened its shell. You can see the inhalant and the exhalant siphon. In this slide, I wanted to make this big point. This is why the water in a lake like Minnetonka or anywhere else becomes clear. Each adult mussel, let's say half an inch long, can take in 1 quart, 1 liter of water a day. and it removes all that very, very fine microscopic plant material and converts it into muscle tissue, and that's why the waters get clear, and that's why your milfoil in Minnetonka will now be more abundant because sunlight goes to the bottom in areas it didn't go before. So I just wanted to introduce a few of those slides for Zebra Mussel 101.
Well, thanks, Dan. And the 1 quart or 1 liter of water per day doesn't sound like much, but when you multiply that times lots of mussels, I've heard accounts where it's been calculated the entire volume of Lake Erie is filtered each day. Is that reasonable?
Lake Erie is pretty big. Those kind of quotes have come from example, like I'm here in New York, right? So the Hudson River, during the peak of the zebra mussel population, because they do go up and down, half of the water in the Hudson River was going through zebra mussels every day. So every 2 days, the entire content of the Hudson River. And as you say, Dick, for a mussel the size of your thumbnail to take in a liter or quart of water, it may not sound like very much, but when you have so many in a lake, that's why the water gets clear, because they're literally filtering out all the fine particles, searching for food.
Yeah. Well, I don't think it's going to take too much convincing at this side of the world that zebra mussels are not a good thing in Lake Minnetonka or in any lake. So let's go on to describe the trials and what they're intended to find out and how that might work.
Yeah, for the inventor of this biological method, for me, this is pretty exciting stuff to have a group of scientists that are top-notch in the USGS, along with their collaborators, including the New York State Museum, where I left a couple of years ago. I went to the University at Albany now. That's where I'm now. To have this team, they've been working together for 2 to 3 years already. And I predict that this 3-year study, and they received $600,000 from the Environment and Natural Resources Trust Fund in Minnesota for it. This is going to be a landmark study in the evaluation of this biologic control method. And I invented it, and it was licensed by New York State to this company in California, and they gave it the product name Zequanox. There's 2 main things that this 3-year project is going to do. It's going to look at the efficacy, how effective it is in a lake, and also it's going to look at the safety of this product, Zequanox, in a lake. And I can go into more detail if you wish, Dick.
I think you should.
Okay. And cut me off at any point. Okay, in terms of the safety, they have put, and let's go to that next slide. It's entitled Samplers That Zebra Mussels Will Settle On.
It's up now.
So it's up now. Okay, good. So what let me just back up a little bit here. What USGS has done, again, with their collaborators earlier this month, in the month of June, is they put these types of samplers, they're called multiplate samplers. You may not be able to tell from the photo, but those plates are mounted on a cement base. I'm going to guess maybe 2 foot square. And they place these in Robinson Bay in Lake Minnetonka. And what they want now, they did this a few weeks ago in June, what they want is for those plates to become colonized. It's about 10 feet deep where they put them. They want those plates to become colonized by aquatic insects and zebra mussels. And then they'll give it, let's say, June, July, August, September. I think it's September. Then they're going to take those samplers and divide them up and put them in different places in Robinson Bay. And then, and I'll show you some photographs on this shortly, then they'll have a group of these samplers on the bottom, and they'll treat so that the aquatic insects and the zebra mussels that are on those parallel plates will be exposed to Zequanox. So it's, for someone like myself, it's pretty exciting that in an actual lake on a naturally colonized plate, we're going to see the efficacy. Remember I said there's two things they're out after, efficacy and safety. We're going to see how effective Zequanox is in killing the zebra mussels that attach to those plates. And we're also going to see if there are any impacts on what we call non-targets. So it would be the aquatic insects that are crawling around on those plates. So those are the two main things they're going to do in terms of it being in the lake and doing studies. They're also going to do studies at their lab in La Crosse, possibly also at the New York State Museum, looking at impacts in very controlled conditions. What we call in a mesocosm, it's basically an outdoor artificial pond. They'll do some studies on the sublethal effects even on fish. The fish don't die, but do they spawn as well? Do they produce as many eggs, etc.? So efficacy and safety, those are the two things that this study is going to do. I just would like to go to the next slide, which shows Robinson's Bay. And there's a little yellow area. That's where they put those samplers a few weeks ago in June. And then again, just want to repeat, in a few months, then they'll divide the samplers up into groups, and they'll treat some, and then they won't treat others. It's what we call an experimental control. So they'll have something to compare to. I'd like to go to the final slide that I supplied, and you'll see a yellow rectangular area, and this is exactly what it's going to look like, let's say in September, when they placed some of these samplers in an area, and when they go to treat, they're going to put Zequanox inside that barrier. And just imagine sheets of plastic going down to the bottom and sealed off around the samplers that are inside there on the bottom. So to be able to treat this very limited area, and it's very difficult to treat lakes when you're trying to get mussels killed on the bottom. So this will allow very controlled conditions. They will be able to inject in some of these enclosures that they have. I'm not sure how many they're going to have. Let's say they'll have a dozen in total. In some of them, they'll inject the Zequanox right to the bottom where the mussels are and the samplers. And then in others, they'll treat all of the water inside that enclosure and see what gives the best kill. So, two things, safety and efficacy. It's very exciting because there have not been many studies at all done in lakes. So definitely, this is going to produce very interesting, valuable data.
Okay, well, Dan, you've been working with the product, Pseudomonas, which is, the trade name is Zequanox, for many years. You know it kills mussels. Why do we need to do this?
You need to do this. Again, you, in a lab, let's go back and just make it real simple. Take a zebra mussel in the lab, put it in a little beaker, a glass beaker, put in some Zequanox. The mussel feeds on the Zequanox and dies. Now, does that mean you can go to an area in a lake and get the same effect? No. Because how are you going to deliver that Zequanox to the mussels that are either on attached milfoil or at the bottom? This is one of the major challenges of trying to go from the lab to field. The field is a lot more complex. So that is going to be a major challenge. Can they deliver the Zequanox in a real situation? Granted, it may be inside that enclosure, but can they deliver it to the mussels? One, the right concentration, and number two is big time. The duration. You can't just treat these mussels for 1 minute with Zequanox and expect them to die. It requires at least several hours. So that's why this study is so important. This is a real life situation. Granted, it's kind of artificial. It'll be inside these enclosures, but that's a step towards one day applying it to, let's say, a beach area in Minnetonka or a dock that wants to have zebra mussels, the majority of zebra mussels removed. So it's a step toward real application, which hopefully will be available. You know, that's why you do research. You find out if it works or not in the field under real conditions.
Okay, so we've heard reports already that we or the researchers have put the samplers in June, and they're going to do the test trials in September, and by the end of September, we'll know whether it works or not, right?
Well, you'll collect that data, and again, hopefully everything, Dick, hopefully everything will go well. But one of the challenges in any time you try to do a field study is called Mother Nature. So if, they'll have these enclosures that have to be there during the treatment. Hopefully there won't be any problem on treatment day. Hopefully after they remove the enclosures, after a certain number of hours, it depends on their experimental design, hopefully those samplers, which are at the bottom, won't be disturbed during the month that they're waiting for all the data to come in. So they may find that they get excellent kill. They may find that they don't get excellent kill. And as scientists, they look at the experiment and they've measured all the different variables and determined either why it did work or it didn't work. Let's look at the best possible scenario. Even if they get incredibly high kill, 98% kill, even if they look at their non-target data, all those little aquatic insects weren't disturbed at all. That's very encouraging, right? But we still have to wait for the U.S. Environmental Protection Agency to issue a registration to Zequanox for use in lakes. Right now, the U.S. EPA has given permission only for Zequanox to be used in power plants, etc. So it hasn't given permission for it to be used in open waters, such as lakes or rivers. So the U.S. EPA has to give permission. They may use the USGS data, to make that decision. So that has to occur before anyone at Minnetonka would be able to apply. And there's another big factor, and I'm going to be very clear. You will never see, whether it's Zequanox or some other control agent, you're never going to see all of Lake Minnetonka treated. The economics of doing that, so the economic feasibility, as well as the technical feasibility. Remember I talked about how do you do to where the mussels are. So you're never going to see that. You're not going to see eradication. People dream about that. With the current formulations of Zequanox, you're not going to get rid of all the mussels in Lake Minnetonka. Now, maybe someday, some other control method, or if they come up with a super-duper formulation of Zequanox someday, you might have a shot at that. But right now, I think people have to be realistic. If everything goes really well, it'll be used to clean off beaches. It'll be used to clean off dock areas after U.S. EPA approval.
Okay, so in the best of circumstances, it's going to be several years before we have some clear answers.
I think so, yeah.
Okay.
Yeah.
Okay.
And those answers, that's the thing, those answers, without studies like this, you're not going to get those answers. That's why I was delighted to see that this grant came in Minnesota, and this very competent team now can go forward.
So would it be fair to say this is the first step in working the problem?
Yeah, it's maybe not the first step. I think, I'm not going to say it's the sixth step or the 10th step. Work has been done. A lot of work has been done on the safety of this control agent, but it's been more laboratory. Many investigators have looked at this bacterium, which is the active ingredient that we found at the State Museum back in the 90s. And we developed a lot of what you call acute toxicity data. Do these different organisms, other than mussels, do they die very quickly? Look, there's a lot of data on its safety. So, we've done a lot of step 1, 2, 3, and 4. This is now getting to open water applications. And that's where we need more data, and the U.S. EPA wants more data.
Okay. So it sounds like we're going to be living with zebra mussels on Lake Minnetonka for a little while yet.
Dick, for generations. That's it. For generations. It's only in limited areas that control might be technically and environmentally feasible. Only in limited areas. You're just not going to have the entire environment of Lake Minnetonka treated. It's just not going to happen.
Okay. We just have a couple minutes left. I know you're aware that Minnesota has recently established a research institute for invasive species to look at other controls for other invasive species. Good move?
From my perch here in New York, and again, I'm a guy who's been working on zebra mussels for over 20 years, I cast a wide net in terms of seeing what kinds of research are being done in different states. And I'm not trying to make you Minnesotans and you Lake Minnetonkaans feel good. This is not a feel-good statement, okay? It may make you feel good. Minnesota is absolutely a leader. The state of Minnesota is an absolute leader in dealing with aquatic invasive species. Now, again, I focus mostly on what states are doing what with zebra mussels. So in terms of zebra mussels, whether it's now you brought up this new Minnesota Aquatic Invasive Species Research Center in St. Paul, in the Twin Cities there at least, I think it's St. Paul, and that establishment last year by the legislature, they provided funds not only for looking at new, inventive, creative ways to control zebra mussels, but also how to deal with carp issues and aquatic plants. One of your favorites, Dick, milfoil and et cetera. So that center is another reason why you look around at different states across America. Has any state legislature established a similar center? I don't know of one. And so there's Dr. Peter Sorensen there, who is the director of it, and I predict some really interesting research will come out of that center, because the people of Minnesota told their legislators how important it is to control invasive species. You brought up the center, Dick, but also you have your DNR, your environmental regulatory agency, is also frequently in the media because they're doing all sorts of things to slow the spread of zebra mussels in your land of 10,000 plus lakes. Whether it's dogs, they've got 3 or 4 dogs that are trained for sniffing, besides doing other kinds of sniffing with their law enforcement officers. You've got, DNR.
Dan?
Yeah.
I'm going to have to interrupt. We're getting the heave-ho. We're out of time.
Okay, but I wanted to definitely tell you the truth about your state. You've done incredibly proactive work.
Yeah. Well, thank you. Thank you, Dan. It's always a pleasure. We'll look forward to seeing you again next time you're in town. And
Sounds good.
And if people have questions, they know how to contact me. That number and email is always flashed. And if they have questions of you, Dan, I'll feel free to have them forwarded your way.
Sounds good. And thank you for the invitation to be on your show. I appreciate it very much.
That's it. Thank you.